Hello, and welcome back to today's lectures where we're going to start taking a look at human gene therapy. And so we're going to start out with somatic gene therapy, and somatic are going to be body cells. So we're going to be looking at kind of repairing the cells of the body using some of the techniques that are very, very similar to what we're talking about changing the DNA, changing the genetics of organisms. And so we've looked at it on microorganisms like bacteria and yeast, and that's a process that's relatively simple. Looked at it a little bit within kind of higher organisms, looking at genetically modified plants and animals. And now we're going to make that transition to look at trying to go through and modify the genetics of human beings. And so what we take a look at is that this concept of gene therapy is the idea that we're going to go in and try to treat diseases, primarily genetic-based diseases, using the same type of recombinant DNA technology that we use to make, genetically modified organisms or genetically modified kind of plants or animals. So the whole idea behind gene therapy is that we're going to identify, what is the DNA, the gene and the protein that's associated with some type of specific genetic disorder or disease. And we're going to go in and we're going to try to fix that defective gene. We're going to try to put in the proper DNA, the correct DNA, so that when that DNA is expressed, it's going to cause for kind of a repaired protein that can act normally within the cell. And so the idea then, again, because we're focusing in on human gene therapy and at least in today's lecture, this idea that what we're going to do is treat the body cells, somatic cells within a human being. And so normally what we're going to do is not go in and actually kind of repair the damaged piece of DNA, the incorrect piece of DNA. We're not going to go in and kind of remove the damaged part and put in the correct part. Normally what's going to happen is we're just going to put in the correct DNA, the replacement DNA or the replacement gene, get that into the cells somehow so that the individual cells may be, producing both the products of the damaged gene as well as that replacement recombinant DNA. And so, it's not going through and fixing the DNA per se, but it's going in and, providing the correct information to the cells that need it. Now, the most common way of delivering this replacement gene, this kind of repaired gene, the repaired DNA, into human cells is to use a viral vector. And so we've talked about viruses a little bit before in the course, and the idea that we can use them to carry some type of nucleotide message, either RNA or DNA, because viruses are really, really good at infecting cells. And when a virus infects a cell, it basically injects in the RNA or DNA that's found within the virus into the cell, and it kind of takes over the cell. It gets the cell to kind of make copies and to express whatever genetic material that it injected in. And so there are a variety of different viruses that are available. And so we've got retroviruses, retrovirus, kind of a backwards virus, if we want to think about it that way, is an RNA virus, like HIV, the virus associated with AIDS. And what happens with this is that it's got RNA within the virus. And so when it's injected into the cell, it has to back up a step. And that's why it's saying a retrovirus. It has to back up and take the RNA, move backwards to make DNA, and then use that DNA to take over the cell. Now, there are going to be other types of viruses as well. There are going to be adenoviruses and herpes simplex virus. Adenoviruses are what we would see kind of spreading the common cold. It's, very good at, infecting human beings and carrying that genetic information. And then herpes simplex virus, these are the viruses associated with primarily cold sores and some other things. But again, it's a virus that's going to be very good, very effective at infecting human cells. And so what we want to do is basically go in and alter these viruses in a way that they're going to deliver our nucleotide message, deliver our replacement gene into the cells, but alter it in such a way that, it's not going to become infectious. It's not going to produce lots and lots of virus particles that are going to spread out and, infect other cells or infect other people. And so mainly when we're using these viral vectors, we're going to go through, and again, like we talked about with the Jurassic Park example, we're going to go through and we're going to ideally fix it so that it's not going to be able to kind of escape and take off on its own. And so these viruses are going to be altered in some way, so they're going to be able to infect the cells, but they're not going to be able to spread. So if we take a look at this, again, the idea is that in gene therapy, what we're going to do, and the best example, at least the first example that we're going to take a look at, is going through and treating cystic fibrosis. Cystic fibrosis is a fatal lung disease. It's associated with a defect in a specific protein within the lungs. And so the idea is that if we can put in the gene, put in the DNA message for the correct version of this protein, the cells in the lungs can produce the right form of the protein, and in doing so, kind of eliminate most of the problems and the symptoms associated with cystic fibrosis. And so the whole idea is that basically you go in, you identify the gene, like we've got over here on the right-hand side of the slide. You're going to find, where the damaged region is, what's wrong with it, and then we're going to take the replacement gene, kind of the corrected version of that message that we want. We're going to package it within a virus, and then take that virus, which, like viruses do, are going to infect cells. And so in this case, you're going to use like almost like an inhaler. So you can deliver these virus particles down deep within the lungs of the patient, with the idea that what's going to happen is that virus is going to infect the lung cell, and infecting the lung cell, it's going to inject in that DNA, inject in that gene, and when that cell expresses that gene, it's going to produce the correct version of the protein, and hopefully, at least minimize the effects of the disease, and put off or delay the onset of some of the more severe symptoms associated with the disease. Now, if we take a look at this, in order for this to work, what needs to happen is that we've got to be able to get the virus to infect the cell. When the virus infects the cell, the DNA has got to be inserted into the cell. And that's, that works. That's what the virus does. But that DNA that's now injected into the cell has to be there and present, ideally integrated in some way that's going to become permanent within the cell. And that DNA needs to continue to be expressed. But what we're going to see is that many times in the patients that are being treated with this type of gene therapy, over time, either the cells stop producing the DNA, stop using the DNA, producing the protein, and that's normally a process that as the cells develop, they kind of change what DNA they're expressing as they're going from a development to a maturing cell. So that may occur. We can talk about the immune system in the next couple slides. So that basically, we can get this corrected message into these cells. We can get these cells producing the right version of the protein, but it only lasts for a period of time. Over time, this kind of breaks down, the process breaks down, so that these patients then have to go into another round of gene therapy. Now, one of the reasons for that is that the immune system, as we've talked about before within the course, is that the immune system kind of attacks cells that are recognized as being different, recognized that are being foreign. And so if we're going to use this viral particle, this virus, to inject in, a nucleotide message, a correct gene, there's the possibility that when that cell is infected and when that cell is expressing that gene, the proteins that it's going to be producing are going to be looked at as being different from the normal proteins and the normal cells within the body. And so the more times that you kind of use this viral to treat, through the process of gene therapy, to treat these cells, you end up mounting a greater and greater immune system response. And so, the cells in our body that are good for fighting off pathogens are going to be looking at those cells that have been infected with the virus and not recognize they're infected with the virus because we're doing gene therapy, but recognize that they're infected with a virus. And in the past, when things are infected with a virus, that's usually bad. That's usually associated with the development of a disease. And so because of that, it's thought, and there's good evidence, that many of the cells that have been altered using this gene therapy are going to be attacked by our immune system. And so they're going to be killed off. And so that's why we have to go through and try to figure out another way to do it, so do another round of gene therapy, or figure out a better way of getting the DNA into these cells and getting these cells to do what they need to do. Now, the viral vectors that we're using are the best for what we've got, but they're not going to be perfect. And the reason for that is, again, because we're using a virus, and the virus causes disease under normal circumstances. So even though we're changing the virus in a way that makes it less disease causing, the body's not going to recognize that. The body's going to go through and attempt to kind of figure out where the virus is and attack the virus as individual viral particles, as well as kind of going through and attacking those cells that have been infected with the virus. And so when we have this gene therapy going on, we're often going to see immune and inflammatory responses, kind of similar to what we would see if we would have like an organ or a tissue transplant. We want to try to minimize the rejection. The other reason that viral vectors aren't perfect, and no vector at this point is perfect, is the idea that it's going to go through and basically infect all of the cells in the region. We don't have a mechanism at this point that we can go through and only target the specific cells that we want to have, in this case, the corrected version of the gene. So, it works. There are problems associated with it, but we're kind of learning from these problems and improving the process as we're moving forward. Now, again, the concept that viral vectors aren't perfect is that they're going to put in that replacement gene, but in many cases, they're not going to have a lot of the control regions that we talked about. We talked about the regions of the DNA that are kind of like on-off switches. And usually there's going to be an on switch associated with the gene that we're putting in. But there are other things like amplifiers. They're called enhancers and things like that within the scientific realm. But they basically mean that we're going to produce lots and lots of this gene. Sometimes we lose that message when we put our corrected gene or replacement gene into these cells. And so because of that, we don't get the same type of control. We don't get the same type of kind of production of the protein that we would get normally within the cell. And so because of that, that again limits the ability of gene therapy to be able to form a long-lasting and permanent treatment for some of these genetic diseases. Now, the other thing is that at this point, we're still at the kind of infancy of gene therapy. So it's only effective at treating single gene disorders. And those are things where, there's a single defect within a gene. And because of that, all we have to do is just replace that, that defective gene or that incorrect gene, put in a correct version in the cell, and everything hopefully is going to be fine. The problem is, even though there are a fair number of single gene disorders that have been identified and we're learning more and more about them, most of the big diseases that we worry about, thinking about some of the things we talked about in terms of the most prominent causes of death within the United States, or the examples of debilitating diseases that have an impact on quality of life and families and society. Things like heart disease, high blood pressure, and Alzheimer's disease have lots of genes that are involved with them, lots of environmental factors that are involved with them, lots of other things that are going on. And so because of that, it becomes more and more difficult to treat something if there are a lot of things that are causing the disease. And so we're at the cusp of being able to treat these monogenetic or single gene disorders, but we're still probably quite a ways away from treating these multiple gene disorders as well. Now, as I said, cystic fibrosis was the first example of human gene therapy, and it's the one that has been most successfully treated with gene therapy up until this point. Now, cystic fibrosis, like I said on the previous slide, is an example of a single gene defect. There's a protein in the cells within the lungs that express, and again, a huge scientific name. It's not that important that you know the name, but just understand that there is a protein that's involved. It's cystic fibrosis transmembrane regulator gene. Was discovered in 1989. It's got lots of different identified mutations in it. And basically what it is going to be a protein that's found along the surface of the cells within the airways. And so what we've got on the right-hand portion of this slide is the airways within the lungs. And so we're going to have these almost like little tubes or tunnels that are going to be going kind of connected to your mouth and your nose to let air go in and go out. But it's going to be kind of directed down deep within your lungs. And so you want the air to be able to flow through these tubes or tunnels relatively unimpaired. You don't want it to be impeded in any way. And so normally what's going to happen is that we're going to maintain a moist surface along the inside lining of these cells. So it's going to be moist along the inside of your body. And the reason for that is that these cells are going to be secreting, or special cells within the airway are secreting mucus. It's going to form this kind of moist covering along it. And it keeps it moist, it keeps it clear, and everything's fine. Now, what's going to happen within these normal cells is that they have a functioning cystic fibrosis transmembrane regulator protein. Again, it's a mouthful in the name, but it allows these cells to pump chloride into this region, into the airway passage. That chloride is like a little bit of salt, and that salt is going to cause water to draw into that region and try to kind of dilute it away. So we're going to have water, which is going to be accumulating along this moist surface here. And in doing so, it's going to continue to kind of dilute the mucus and keep the mucus from building up. Now, in a patient with cystic fibrosis, they're not going to have that cystic fibrosis transmembrane regulator protein. And so they're not going to be able to pump the chloride across the surface. They're not going to allow, kind of the movement of water to kind of help maintain kind of this moist surface along the way. And they're not going to have that water to dilute the mucus. And so the mucus is gradually going to build up. And so think about, if you've had seasonal allergies, you've got a head cold, you know what it's like when mucus is building up within your air passageways. Think about, all of the, for lack of a better term, the non-scientific version of mucus is snot. Think about all the snot, all the mucus that's being produced in your nose, and all of that, kind of gloms down, it runs out, out your nose, down your face. It's going to be running down through the back of your mouth. But think about that mucus now down deep within the air passageways, these tubes and tunnels. They're going to be relatively narrow down within your lungs. And now they're glommed up with a bunch of snot. They're glommed up with a bunch of mucus. And that's going to make it very, very difficult for the air to pass through it. It's going to be a location where particulate matter and things like bacteria can start to accumulate. And in doing so, it's going to impair, make difficult the movement of air. It's going to increase the risk of bacterial infections. It's just going to cause all kinds of different problems, all because we got a single protein deficit, which allows us to, or prevents us from pumping water across the membrane and diluting out this mucus. And so with the single gene defect, cystic fibrosis transmembrane regulator gene and cystic fibrosis transmembrane regulator protein, without that, this is what builds up and this is what causes the damage to the lungs of a patient with cystic fibrosis. Now, cystic fibrosis is a fairly common fatal disease affecting about 30,000 people within the United States. If we take a look at how it's going to be passed, it's estimated there's about 1 out of 31 Americans is a carrier. And so that basically, within the United States, and this number is a little bit old now, but there's probably 10 or 11 million carriers of the cystic fibrosis mutation out there in society. And what happens is if you have an unaffected carrier father, an unaffected carrier mother, they've each got this kind of damaged portion of the gene, this incorrect portion of the gene. But because they have the functioning gene, a copy, remember, humans are going to have 2 copies of the genes in the ones that we're looking at. We're not going to talk about sex genes at this point. But they're going to have 2 copies. So if they got 1 copy that's correct and 1 copy that's damaged, that's enough. their one copy that works is going to be sufficient to keep the cells working properly and keep the lungs healthy. But what happens is that 1 out of 4 chance that a carrier mother and a carrier father are going to produce a child which is going to have both copies, which are going to be that defective, incorrect information. And that's going to give rise to about 2,500 to 3,000 newborn infants per year. And again, in these patients, they're missing that protein. They're unable to dilute the mucus within the lungs, dilute the snot, in essence, within your lungs. And so because of that, they're going to have difficulty breathing and be very, very susceptible to lung infections. Now, in 1993, relatively soon after they discovered the protein and began able to understand what was going on, they started to do gene therapy to treat cystic fibrosis. And so they used a common cold virus as a vector. And so like we talked about before, we got this kind of common cold virus up here. We're going to put in the correct version of that gene, so the cystic fibrosis transmembrane regulator protein, and this, we're going to, or gene, we're going to put in the cystic fibrosis transmembrane regulator gene into this virus. So we're going to get the virus inhaled by the person. Hopefully it's going to get down deep within the lungs, so that when we look at the lung cells over here, what's going to happen is the virus is going to infect the cell. It's going to deliver that correct replacement gene. It's going to be expressed by these cells, and now the cells are going to be, in essence, producing that protein. And in producing the protein, they're going to cause water to kind of accumulate in this region, and in doing so, dilute the mucus away. And so it is an effective way of treating cystic fibrosis. But like we talked about a couple slides ago, this is something that, you know, either these cells stop producing the DNA and producing the protein, or these cells may die, they may be attacked by the immune system. So we're able to treat cystic fibrosis for a period of time, but then we have to redo this. We have to do another round of gene therapy. And so in about the 30 years since we started doing gene therapy to treat cystic fibrosis, we're now looking at a whole variety of different delivery methods to go through, ideally to have better integration of the DNA, better expression of the protein, and less need to have continual treatments going over and over and over again. Now, an example of this is in 2015. We've got a patient over here in Great Britain. Instead of using viruses as the main mechanism and inhaling those, what they did is that they delivered the gene therapy using viruses in fat droplets. And so basically, they kind of masked the virus a little bit within the fat droplets. They put it in a nebulizer, put it in almost like an inhaler here, so that the patient breathes it in, and it appears as if they're able to, again, get these corrected versions of the gene into the cells that need it, and hopefully without a lot of the immune system problems that were going on before. It appears to be effective in treating cystic fibrosis. It's made it to the phase 2 clinical trials and as of 2015. And so, you know, it appears to be doing good things. The problem is that, like many of the treatments that we're going to be talking about, they're not equally effective in all patients. And so some patients are going to respond relatively well to this treatment. Other patients are going to have more difficult. And this finishes up the first half of this lecture. And so what we're going to do in the second half is continue looking at the history of gene therapy. As always, if you have any questions, feel free to email me. Thank you and have a great day.